All right, let's jump right in. We're about to connect something you see every single winter to the tiny microscopic cells that literally make you, well, you. We're going to uncover the hidden power of solutions. You've seen it a million times, right? The salt trucks are out covering icy streets. But have you ever really stopped and wondered, what's going on there? I mean, how can a handful of simple salt crystals defeat a solid sheet of ice?
A lot of people think the salt must be creating heat, kind of like one of those chemical hand warmers. But nope, that's not it at all. The real reason is way cooler, and it's all about a beautiful little trick of fundamental chemistry.
So, let's break down the mechanics. Why exactly does salt melt ice? The answer is all about how dissolving something can completely change the rules for a liquid. Okay, so to get this, we need to know two key players, the solute and the solvent. The solute is the stuff that gets dissolved, that's our salt, and the solvent is the thing doing the dissolving, in this case, water. Now, when you mix them, you're not just making salty water, you're creating a brand new solution that plays by a whole different set of rules. And this is where it gets super interesting. The properties that change are called colligative properties. And here's the most important part. The solution doesn't care what the particles are. It could be salt, it could be sugar, doesn't matter. All that matters is how many particles are swimming around in there. It is purely a numbers game. Basically, the solute particles just get in the way. This quote here explains that fewer water molecules can escape into the air as vapor. And the exact same thing happens with freezing. The salt particles are like a rowdy crowd, making it way harder for the water molecules to neatly line up and form an organized ice crystal. That disruption, that's the whole secret. This graph shows it perfectly. See that pink solution line? Notice how it's pulled down lower than the blue pure liquid line? In chemistry, that's called lowering the chemical potential, which is really just a fancy way of saying the solution is more stable, more chill. It doesn't want to change. And because it's more stable, you have to make it even colder to force it to freeze, or even hotter to force it to boil. So this one single principle gives us two really powerful effects. Freezing point depression, well, that's why salty water can stay liquid even when it's below 0 Celsius, and that's what melts the ice on the roads. And on the flip side, boiling point elevation is why adding salt to your pasta water actually makes it boil at a slightly higher temperature. Okay, so let's build on that idea of the crowd. If this whole effect depends on the number of particles, what makes one crowd more effective than another? Well, the first thing is pretty intuitive, right? The more stuff you dissolve, the bigger the effect. The more particles you pack into that solvent, the more disruption you get, and the bigger the change in the freezing and boiling points. A little salt helps a little, a lot of salt helps a lot. Simple as that. But, and this is a big but, not all solutes create the same size crowd, even if you add the same amount. This is where chemists use a little secret weapon called the Van 't Hoff factor. We just call it I. And it's basically a multiplier that tells us how many effective particles you get for each molecule you dissolve. And this is the perfect example. When you dissolve sugar, each molecule just kind of floats around by itself. So its I factor is 1. Pretty simple. But when you dissolve table salt, sodium chloride, each and every unit breaks apart into 2 separate ions, a sodium ion and a chloride ion. So for every 1 particle of salt you add, you get 2 particles causing trouble in the water. Its I factor is 2, which makes it twice as good at melting ice as sugar is, molecule for molecule. So understanding this is one thing. But then, scientists realized they could flip this whole idea on its head and use it for something truly brilliant. It became a clever lab trick. Think about it like this. If we know for a fact that the drop in the freezing temperature is tied directly to the number of particles, what if we have some mystery substance? Could we dissolve a known weight of it, measure that temperature drop, and then work backward to figure out the mass of one of its molecules? And the answer is a resounding yes. The process is called cryoscopy, and it's just so elegant. First, you take a pure solvent, say water, and measure its freezing point super precisely. Then, you weigh out a very specific amount of your unknown stuff and dissolve it in there. Then, you measure the new, lower freezing point. And from that one change in temperature, a simple formula lets you calculate the molar mass. You're basically weighing molecules you can't even see. And this isn't just some textbook idea. This is a real technique. Scientists use this specialized tool called a cryoscope to do exactly that. It lets them get incredibly precise measurements of that temperature change. And for more than 100 years, it's been one of the classic ways chemists figure out what an unknown compound is. Okay, so we've gone from icy winter roads to the inside of a chemistry lab. But now we're going to take the biggest leap yet, because the same fundamental principle isn't just a curiosity, it is literally the pressure that drives life itself. See, in biology, this effect gets a special name, osmotic pressure. It's the exact same idea, water wanting to move across a membrane from a less crowded area to a more crowded area. And this force is absolutely everywhere in living things. And its influence is just staggering. I mean, on the most basic level, osmotic pressure controls the very architecture of our cells, like how big the nucleus is compared to the rest of the cell. It helps organize the cell membrane. It's crucial for making sure cells can divide properly. It even helps tell a stem cell what kind of cell it should become when it grows up. From the unique shape of a red blood cell to the way a neuron fires, this simple pressure is the hidden force pulling the strings. And this isn't some tiny, obscure field. Get this, a single review paper from 2024, just on the biological role of osmotic pressure, cited 166 other scientific papers. It is a huge, active area of research, which just shows you how central this idea is to understanding life. So think about where we ended up. We started with something so ordinary, salting an icy road. And by just pulling on that one little thread, we traveled through basic chemistry and landed at the very core of how living things are built and run. The same force that clears our roads also builds our bodies. It really makes you wonder, right? What other invisible chemistry is out there quietly shaping our entire world, just waiting for us to notice?